In this recording, we're going to discuss epithelial tissues. We can find epithelial tissues on every internal and external body surface that we have. They act as barriers between our body and the external environment, and they also line organs and fluid-filled cavities. Some basic functions of epithelial tissue include protection from both mechanical and thermal thermal injury. Okay, I guess you could be thermal energy too. Thermal injury. Your epithelial tissues can also play a role in your immune defense, specifically acting as a physical barrier. So as long as your skin, for example, is nice and intact, it's harder for bacteria to penetrate into our bodies. We can have secretion occurring like sweat, hormones, even the oils on our skin. The hormones get secreted into the blood. The oils and the sweat are secreted through ducts. So we have several different ways of performing secretion. We can transport into other tissues. So a lot of our epithelial tissues are very good at diffusion because they act as a selectively permeable membrane. And then we have sensation. So a lot of your epithelial tissues are very rich in nerve supply, and so we can detect changes both internally and externally in the environment. The ones that detect external stimuli, those are a little bit easier for us to recognize. So if, if a fly or a bee lands on your arm, it's easy for us to feel that because it's also easy for us to see that. So we have multiple ways to acknowledge that, oh, there's a bee on our arm. But internally, it's a little bit more difficult for us to consciously be aware of those changes, but subconsciously we know things are changing and we can mount responses to those changes. So your epithelial tissues are packed tightly. Okay. You have little to no visible extracellular matrix. That doesn't mean it's not there. It's just there's so many cells and they're packed so closely together that you can't really see it. One of the ways that we accomplish this is through tight junctions and desmosomes that we've previously mentioned. So in our picture, we've got those tight junctions at the top of these epithelial cells. And remember, that's going to prevent things from moving in between adjacent cells. And the desmosomes are closer to the bottom of these cells. And we talked about desmosomes provide extra strength by holding these adjacent cells together. Epithelial cells tend to arrange themselves in sheets or layers, okay? And these layers are fairly impermeable. They are resistant to physical stresses as well as mechanical injuries, especially if you have multiple layers. Okay, so your skin, for example, you have several layers, and it does a pretty good job of being resistant to mechanical and physical stresses. Your epithelial tissues are avascular. They do not contain direct blood supply. So notice our image, our epithelial layer is up top. There are no blood vessels. We do have blood vessels down below in the connective tissue layer. This is a very common arrangement, an epithelial layer on top of a connective tissue layer. So because your epithelial tissue does not have direct blood supply, that actually causes something very important to happen. It limits their thickness. Okay, so why do we need blood? We need oxygen and we need nutrients. So your epithelial tissues do not have as easy access to the oxygen and the nutrients they need to stay alive, which means anything that's really close to the blood vessels, so this cell layer right here, this would be close enough to do diffusion. Okay, so we could get oxygen, we could get nutrients, we can stay alive. If we continue to build on top of this one layer At some point, we're going to be so far away from these blood vessels that diffusion is just not going to cut it anymore. But if you don't have supply to the oxygen and the nutrients, you're not going to be able to survive. And so your skin, I keep talking about your skin having multiple layers. The very top few layers of your skin, they're actually dead cells because they're so far away from this blood supply that they just can't survive. Now, your extracellular matrix that we've mentioned is found beneath the basement membrane. Okay, so we've got basement membrane down here. Okay, it's at the very bottom. These are the epithelial cells, and then we've got that little bit of extracellular matrix. Okay. When we talk about epithelial tissues, there are two terms that we throw out a lot: apical surface and the basement membrane itself. The basements are in the bottom of a house, so your basement membrane is the bottom layer. This also happens to be where your newest cells are created. As new cells are created, the old ones are pushed up, and the very top layer is your apical surface. This would be where your oldest cells are. And again, if we go back to your skin specifically, all of the cells at the apical surface, not only are they the oldest, but they've gotten so far away from that blood supply that they're actually no longer living as well. So when we talk about epithelial tissue, we talk about apical surface and basement membrane a lot. Now, when we name epithelial tissues, okay, so remember, epithelial tissue is our general or our primary tissue name. And then we have subcategories. We've got specific epithelial tissues, and we name them in two ways: by the number of layers and by the shape of the cell. So this one sounds kind of silly, but you can either have one layer or you can have more than one layer, and that's kind of obvious, right? If you have one layer, you are termed a simple epithelial tissue. If you have more than one layer, you are termed stratified. Okay, so we can have one layer, we can have more than one layer. Okay, sometimes it's lots and lots of layers, but sometimes the stratified tissues are usually only 2 layers. Okay, which is why we say one or more, or excuse me, more than one is stratified. Okay. And then we have to also do the shape of the cell. And we have 3 options. 2 of these are pretty self-explanatory. Cuboidal cells, they're short and fat like cubes. Not too bad, right? Columnar cells are tall and elongated. They're tall and skinny like a column. That's not so bad. And then we have squamous cells. The word squamous is not super useful right off the bat, like a cube or a column, but these are flattened. I think they look like little fried eggs a lot, with that big old nucleus being the yolk. But that's just my opinion. And one thing to keep in mind is we need both of these terms. Okay, we need to mix and match. You have to mention the number of layers, and you have to mention the shape of the cell every time you mention an epithelial tissue. So you can have simple squamous, but you can have simple cuboidal, you can have simple columnar, you can have stratified of each of these as well. And so you can't just say, oh, that's simple epithelial tissue. Okay, well, what kind of simple epithelial tissue? Is it squamous? Is it cuboidal? Is it columnar? We have multiple options. So when you name these things, we need both terms. So simple epithelial tissues, these are 1 layer thick. These are really good at transporting substances between different tissues. Okay, these a lot of times, not always, but a lot of times will have additional components like microvilli, which are good for increasing surface area for absorption, or sometimes they have cilia on them, which are good for moving other substances through organs. Okay, so not every simple epithelial tissue will have the microvilli or the cilia, but it is something that you might come across. Okay, and those are the functions. The increasing surface area for microvilli and cilia are good for moving substances. So simple squamous, we have the 1 thin layer of cells, the little fried eggs, if you will. These are really good for rapid diffusion. Okay, so this is very skinny, so you could see where if you've got oxygen in your lungs on this side, it wouldn't take a whole lot of effort to diffuse right into the underlying tissue, okay, and vice versa, carbon dioxide. Okay, you can see we find these not only in the lungs, but parts of your kidney tubules. These also line your blood vessels. Okay, so rapid diffusion, rapid transport, and we've got a couple locations here. Now, simple cuboidal epithelial tissue. Again, we've got our one layer, but now we have cubes. Okay, so our picture here, you're like, oh, cubes, I see circles. Okay, we have to look a little bit deeper. These circles that you might see are ducts, and each of these ducts is composed of individual cube cells. Okay, look at these, like this is pretty cube-like, right? This is pretty cube-like. Some of these aren't quite as cubey as you want them to be, like this is not the normal box shape, right? But sometimes the easiest way to determine the shape of the cell is process of elimination. Okay, so if you looked at this picture, okay, and you realize, okay, each of these is an individual cell, well, what do I have going on here? Because this guy looks funny. I'm like, okay, well, are these really flat like fried eggs? No, not really. Are they each of them tall and skinny like a column? No, not really. Then what's our only option? Our only option is cubes. Okay, and again, just because this one cell is a little misshapen, a lot of times that happens when you prepare slides. Things get a little mushed sometimes. So we do have to look at the overall. What do we have going on overall? And these down here especially really do look like little cubes. These are also good for rapid diffusion as well. It's still pretty easy to take something from a lumen and diffuse it across one cell into, a blood vessel or something like that. You find these a lot in your kidney tubules. You do find these in your respiratory passages as well. You also have several glands like your thyroid gland that make use of simple cuboidal tissue. Simple columnar epithelial tissue, we've got our one layer of columns. These, a lot of times, but not always, are the ones that either come with microvilli or cilia. Okay, again, it could be, doesn't have to be. So if you don't see microvilli or cilia, you don't have to automatically rule out columns. Okay, these are also found in places that do a lot of transport. The microvilli specifically, we talked about microvilli increasing surface area for absorption, so you'll find these in places like your digestive system where we do a lot of nutrient absorption, or you'll find these with the cilia that propel substances, so you'll find these, for example, in other parts of your, like your small intestine, okay, or your uterine tubes, okay, so uterine tubes help propel little eggs through the uterine tubes towards the uterus, okay, so microvilli are good for absorption, cilia are good for movement, okay, so they do look very similar, okay, in appearance, but their function is quite different, okay, and so you can kind of see these little fuzzies at the top. Now, one more thing to keep in mind with simple columnar, notice these nuclei, okay? These tend to be very uniform, okay? Very, I can't think of the good word right now, but they tend to make one nice, simple line, okay? Everybody's kind of uniform, and I point that out because of our next slide. Pseudostratified columnar epithelial tissue, which is a mouthful. These appear to be multiple layers because the nuclei are kind of just all over the place, okay? But pseudo-stratified, pseudo tells us that it's false stratified. It's not really multiple layers. It is technically, I don't know why that was hard to say, technically only one cell thick, okay? Every one of these cells, and again, it's really hard to tell, but every one of these cells comes into contact with the basement membrane, okay? But if you look at these nuclei, you're like, oh, that's like 3 or 4 layers right there. And again, I pointed out that the simple columnar, you've got one nice line, okay? It's very easy to tell that these are just one cell thick, okay? So this one's a little bit harder, Okay, but when we distinguish, we're gonna look for the nuclei. One nice line, not one nice line. Okay. You find these also in your respiratory tract, your nasal cavity. These tend to be ciliated, so we're moving things through your respiratory tract. These are little cilia. You also have goblet cells a lot of times. These pretty good sized cells. They tend to stain more clear. They produce mucus. Okay, so anywhere that you have the need for mucus, both in your respiratory and your digestive tracts, you're liable to see goblet cells. Now, we did not see goblet cells in this particular example of simple columnar epithelial tissue, but you can find goblet cells. Remember, we're in your digestive tract here as well, and we need a lot of mucus there to prevent friction. So that's not necessarily a distinguishing characteristic of simple versus pseudostratified columnar. Okay, just because they have goblet cells. What else are we looking for? We're really focusing on that nucleus if we're trying to compare those two types of tissue. Stratified epithelium. So we've got our first official multi-layered tissue. This is really good for protective barriers. Okay, so you find these anywhere you have a lot of mechanical stress. And these come in two varieties. You have keratinized, stratified squamous epithelial tissue, and then you would also have non-keratinized, stratified squamous epithelial tissue. So when we name stratified tissues, stratified epithelial tissues, we have to make sure we are always looking at the apical surface when it comes to naming the shape of the tissues. And here's a good example of why. So remember, our apical surface is at the top. We've got our little cartoon, we've got our real image as well. And our basement membrane is at the bottom. Okay, our basal cells are going to be at the bottom. So if we looked at the bottom when we went to name these things, some of them have some interesting shapes, but we don't really see boxes, we don't really see columns, we don't even necessarily see flat little eggs because everything down here is so squished. Okay, all of the layers above are just pushing down on these cells at the bottom, and it makes it a little hard to determine the shape. If we look at the top though, okay, it's very easy to see that these are good old squamous cells. Okay, and let me see if I have an example. So for example, we'll skip ahead to stratified columnar real quick. It's very easy to tell that this apical surface, these are columns, okay, these are columnar cells. If you were to look at the basement membrane instead, these look a lot more cuboidal because they're being squished by the upper layer. Okay, so when we name these stratifieds, we're always looking at the apical surface. Okay, all right, let's go back real quick. All right, so the keratinized stratified squamous. We've got our multiple layers. We're going to look at the apical surface for the shape. We've mentioned previously that epithelial tissue is avascular, so everything up here at the top, really far away from the blood vessels, those are going to be dead cells, so they don't have nuclei or anything like that. They are going to be filled in this case with a protein called keratin though, which is what the keratinized part comes from. That keratin protein makes your tissues tough and more resistant to friction. Okay, this makes it well adapted to be the outer layers of your skin. Okay, so that keratinized, that keratin protein, okay, that's the outer layer of your skin. That's where we're going to find this. Now, we mentioned we also have non-keratinized. Okay, so non-keratinized, obviously we're not going to have that layer of keratin. Okay, so what's going on here? This is a little bit different. Your apical cells still retain your nuclei. They're still alive in this case. That's interesting. Okay, so they still have enough ability to gain oxygen and nutrients. These are located on the insides of your body. You're still subjected to mechanical stresses, okay, but you find these tissues, this specific type of tissue, anywhere that you have mechanical stress, but it also needs to stay moist. So your mouth, your throat, your esophagus, anus, vagina, okay. So you can still see multiple layers here. Okay, we're not going to look at the basement membrane for the shape of the cell. We're going to look at the top. Okay, these nice flat little cells. Okay, so we don't see that clear layer. Okay, so this was our keratin layer up top here. We don't have that here. These are still fully nucleated living cells. Okay, so that's keratinized and non-keratinized. Stratified cuboidal epithelial tissue, these are pretty rare. These tend to be maybe 2 to 3 at the most cells layers thick. These line your sweat ducts. Okay, so I mean, I know if it's really hot outside in the middle of July, you're sweating a whole lot, you probably think you have tens of millions of sweat glands, but that's not really the case. You really only have a handful of places that you find these cuboidal stratified ones. Same thing with stratified columnar. These are also rare. These tend to also only be about 2 to 3 layers thick. We pointed out a second ago that this is a really good example of why we always name the apical surface. Okay, these are very clearly columns. The basement membrane would lead us to maybe believe that they were cuboidal cells instead, so we're not going to make that mistake. If you have a male urethra, this is where one of the places you would find stratified columnar. If you don't have a male urethra of your very own, you do probably have corneas in your eyes, and you have salivary gland ducts as well. So you do still have these in your body, more than likely, even if you don't have a male urethra. Now, transitional epithelial tissue. Now this one is a little bit different. We don't say simple transitional, we don't say stratified transitional, we just say transitional epithelial tissue. And these only live in a very, very particular place, your urinary system. They line portions of your kidney tubules, okay, portions, your renal pelvis, your ureters, the bladder, and the urethra. These look a little bit different. The bottom layer, the basal layer, these are a little bit more cuboidal in appearance, a little more boxy. But the apical surface, they're quote-unquote dome-shaped. I say they look like little balloons because they're very round. Okay. Now, the location of these is very important. It goes along with the function. Your bladder, you know, is really good for holding and then eventually releasing urine. When these tissues are relaxed, this is what they look like. They're very round, balloon shaped at the apical surface. If your bladder is full and it's all stretched out, then these cells will also stretch out and they look a lot flatter. Okay, so depending on if you're looking at an example of a full versus an empty bladder, these will actually look a little bit different visually. Now, we're not trying to trick you here, and so anytime we give you a picture of transitional, they're always just going to be these little balloons. Okay, this is also one of the reasons why we look at the apical surface. So we mentioned the more cuboidal appearance at the base of the membrane, but also if you continue to look farther down, okay, if you go past the basement membrane, now you're into connective tissue here. Same thing here, connective tissue underneath. Same thing here, connective tissue underneath. That's a repeating theme. You'll have epithelial tissue on top of connective tissue or muscle tissue, something like that. So you really want to take notice of when that transition happens. Okay, so this would be our transition point. I know that's coincidental. This is transitional epithelial tissue. But if you go too far down and you start looking way down here and trying to name these tissues, it's going to be difficult to impossible because this is no longer epithelial tissue. This is not connective tissue, okay? So make sure you know where your cutoff point is.